I'm Dr. Chris Masterjohn of chrismasterjohnphd.com, and I'm here with Alex Leaf of Examine.com, and this is episode 58 of Mastering Nutrition, where we talk about why you should manage your riboflavin status and how to do it.
This is Mastering Nutrition with Chris Masterjohn. Take control of your health, master the science, and apply it like a pro. Are you ready?
Hey everyone, in this episode, I sit down with Alex Leaf. Alex Leaf holds a master's degree in nutrition from Bastyr University. He's a primary researcher at Examine.com and part of the teaching staff at University of Western States for their Human Nutrition and Functional Medicine program. You can reach him at his website, alexleaf.com. Alex and me are going to flesh out the series I started and never finished on measuring and managing nutritional status. And basically for the next 20 weeks or so, I'm going to be intensively investigating and updating my views on a particular nutrient. And Alex will part-time do some intensive research specifically into supplementation, and we'll sit down and talk about it. So in this case, we're talking about riboflavin, and I do the bulk of the talking here, but Alex gets to talk for maybe a half hour, mostly on specific questions about the practicalities of supplementation and looking at the supplementation trials that have been done. In the context of doing this research and recording the podcast, I changed my mind about three things. The first is that I went into it believing for years that there was some utility in taking flavin mononucleotide or FMN supplements, also known as riboflavin 5-phosphate supplements, the coenzyme form of the B vitamin, as opposed to just plain old riboflavin, for cases where people had impaired activation of riboflavin to the coenzymated form. And I now believe that it probably doesn't matter, except for people with small intestinal damage. They may actually do better with the plain old, unfancy, cheaper riboflavin supplements. The second is that in testing nutritional status, the ultimate cheat sheet, I included HDRI's erythrocyte glutathione reductase activity test as a marker of riboflavin status. And in the next edition, I'm going to withdraw that, and I'm going to include two different ways of using LabCorp's whole blood riboflavin test. One is going to be to just take it at face value and use their range. And the other is going to be to put it through a specific calculation of mine that I think might be better. And I'm going to put that calculation in the show notes of this episode, as well as in the next version of the cheat sheet, and collect some data and responses from people about what seems to work better to explain their symptoms and help them in actionable strategies. The third thing is, I realized at the moment that Alex was talking about it in this podcast that in my own MTHFR protocol, I have been nowhere near as attentive as I should be to the place of riboflavin. And in fact, I now believe an argument can be made that there is no such thing as a bad MTHFR in the general population in terms of the polymorphisms that everyone's talking about when they say, I have MTHFR. It may well be the case that these polymorphisms only matter on the background of bad riboflavin status, which may be the norm in our population. So here are the cliff notes. Some signs you might not have enough riboflavin include doing poorly on a high-fat diet, having iron deficiency that doesn't respond as you would expect to iron supplementation, cataracts, elevated homocysteine, oxidative stress causing wear and tear on your tissues and making you age faster and less gracefully, high blood pressure, migraines, fatigue, exercise intolerance. In pregnancy, preeclampsia. This is all in addition to the more classical signs that include a moist, scaly, itchy, painful, greasy skin around the mouth, the nose, and the genitals especially, conjunctivitis, crusty lips around the edges and the corners, redness inside the mouth, and in the hands and feet, an increased sensitivity to touch, pain, and heat. Riboflavin's most prominent roles are in energy metabolism, antioxidant defense, methylation, detoxification, and then in a rather large but scattered assortment of roles in other enzymes. Burning fat requires almost twice as much riboflavin as burning carbohydrate. You or your food being exposed to light can increase your need for riboflavin. You spending more energy, like exercising more, can do the same. Low thyroid and adrenal hormones can impair your utilization of riboflavin and your retention of riboflavin. The average person who doesn't eat organ meats and has their diet focus on sugar and fat probably doesn't get enough riboflavin. But so does the person who moves away from the refined, fortified grains that constitute the riboflavin in many people's diets as a major source, and towards high amounts of fat that don't have any riboflavin. Many health-conscious practices that even I recommend around sunlight exposure may increase your need for riboflavin. Alcoholism, various disease states, poor digestion, and bad gut health can cause major hits on your riboflavin status. Riboflavin deficiency is thought to be very rare. It's not a nutrient of concern, but there's evidence that suboptimal riboflavin status is the norm. Part of it is that we just don't consume a lot of riboflavin relative to what our ancestors would have consumed, particularly if our ancestral diets focused on animal foods. And there are other nutrients like magnesium that are needed for riboflavin retention. We cover why most people may actually need 2 to 5 or maybe even a little more milligrams of riboflavin a day instead of the RDA of 1.3 milligrams for the adult male. And we talk about supplementation trials using hundreds of milligrams per day, talking about why migraines and genetic disorders that are very rare are the 2 major applications now, but why there might be a lot of other applications for high-dose riboflavin on the horizon. We talk about when to take riboflavin, how to spread it out, whether to take it with meals, and all kinds of practical details like that at the end. If you would like the details, then listen on right after this brief word from my sponsors. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com. Ancestral Supplements, putting back in what the modern world has left out. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes, 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. All right, welcome back, everybody. I'm Chris Masterjohn from chrismasterjohnphd.com, and I'm here with Alex Leaf from examine.com. Alex, you want to say hi?
Hey, everybody.
All right, so let's start out by considering the parallel stories of 3 people. We will start with the story of Jack. Jack has been out going to the club every night for the last 3 weeks. He has drank a lot of alcohol. He has had a lot of sex with multiple different partners, but he has not eaten, possibly for the last 3 weeks. And he's itchy. He's itchy around the outside of his nose. He has crusty skin developing on the outside of his lips. And his balls itch. His scrotum is getting itchy. And it's kind of greasy. And he's thinking maybe he has an STD. He comes in to see the doctor. And the doctor says that he needs to stop drinking. He needs to start eating food. So he goes back to his usual diet of hamburgers. And he should take a multivitamin. Before you know it, within a couple of weeks, everything starts clearing up. Meanwhile, let's consider a very different story of Jane. She was born named Jane, but she's changed her name to Clover because she prefers to think of herself as a sweet-smelling flower. She drinks a milk diet, completely milk, no solid food, and she's really into sunbathing. She sunbathes for a couple hours a day. She drinks milk exclusively that she has fermented into kefir, but she's fermented it outside in glass jars so that they can absorb, so that the milk can absorb more light from the sun and, take advantage of the energy of the sun. She has very similar problems. She also has itchy, oily skin. It's also concentrated around her lips. The corners of her mouth are starting to break apart. And similar to Jack, she has genital itching, red, greasy, flaky skin. But here it's developing around her labia rather than, in Jack's case, his scrotum. But otherwise very similar. She has a friend who tells her to start eating raw liver instead of just staying with the raw milk. As she does, within weeks, everything starts improving. Now let's consider a third story, Jacinta. Jacinta's story is quite different. She doesn't have major problems with the skin, although maybe it's a little itchier than she'd like. But her main problem is that ever since her pregnancy, she's felt very weak. And her blood work shows that she's anemic. But she's tried iron supplements that the doctor gave her, and not only do they not make her feel better, and not only do they leave her a little constipated, but they're not really improving her iron levels. So she's switched to other iron supplements. She's tried liposomal iron supplements. She's tried heme iron supplements. Supplements designed to have better absorption without causing the side effects, and yet nothing's really moving it. Until she decides to start eating liver. Lo and behold, even though the iron she's getting from the liver is a lot lower in its amount, eating the liver every day is actually moving her iron levels up. So what do these three people have in common? Well, they are all completely fabricated stories of riboflavin deficiency. In riboflavin deficiency, we can look first at the severe complication. So in the classical deficiency state, where you have very severe problems from a very low intake of riboflavin over time, we have uroorogenital syndrome. This is red, scaly, itchy, painful, greasy skin that affects the smile lines, the outer edges of the nostrils, the ears, the eyelids, the scrotum in males, and the labia majora in females. This is characterized by seborrheic dermatitis, which means that the skin is inflamed, and seborrheic just means that the overproduction of sebum makes it an oily or greasy dermatitis instead of a dry dermatitis. The hair follicles can accumulate sebum in little tiny bubbles. In some people, they can get the appearance of what's called shark skin, but technically is called dysebasia. The eyes can become red, like pink eye. They can become teary, and they can become photophobic, meaning increased sensitivity to light. In the lips, we have cheilitis and angular stomatitis. The cheilitis is inflamed red crusting of the outer edges of the lips, and the angular stomatitis is the same thing happening at the corners of the mouth, and when that gets really bad, it can turn into fissures. And these lesions can sometimes be infected with candida. Inside the oral cavity, inside your mouth, you have glossitis, which is inflamed tongue, and that's called magenta tongue because it takes on a magenta color. You have hyperemia and edema, which means that the inside of your mouth can be red, bloody, or swollen. And for blood work, you would see normocytic, normochromic anemia. That means a decreased red blood cell count, but a normal cell size. So if you're looking at a complete blood count, a CBC, the mean corpuscular volume, or MCV, would be a measure of the red blood cell size that tends to elevate in deficiencies of folate or B12 because the cells are bigger than they should be. But in this case, in the normocytic, normochromic anemia of riboflavin deficiency, you have a normal MCV and you have normal hemoglobin concentrations, but you have low numbers of red blood cells. This is due to bone marrow hypoplasia, meaning low content of cells in the bone marrow. And so you can see it involving more than just the red blood cells. You could also see a low reticulocyte count, low platelets, and low white blood cells. Finally, peripheral neuropathy accompanies the classic riboflavin deficiency syndrome. And peripheral neuropathy is a general term for neurological things that go wrong in the extremities, in the hands and feet. Often it's weakness, numbness, pain, or tingling. In the case of riboflavin deficiency, especially likely signs are an increased sensitivity to touch, increased sensitivity of pain, and increased sensitivity to temperature in the hands and feet. Based on what we know about how riboflavin works in the body, and based on some of the studies with supplementation that Alex is going to talk about later, we could also say that for suboptimal riboflavin status, moving beyond the clinical deficiency syndrome, we might also expect to see having difficulties on a high-fat diet, iron deficiency anemia that responds poorly to iron. So even though riboflavin deficiency causes its own anemia, riboflavin deficiency also hurts your absorption of iron and your utilization of iron. So you can have something that looks very much like iron deficiency, might even really be iron deficiency, and yet the ultimate cause is your poor vitamin B2 status. Cataracts can accompany riboflavin deficiency, and although it is not settled and it's currently controversial, it might be the case that suboptimal riboflavin status underlies cataracts in general in the general population. Elevated homocysteine, oxidative stress, which we could think of as the wear and tear on our tissues that increases with age and increases with exposure to toxins like alcohol or cigarette smoke or environmental toxins. High blood pressure, migraines, fatigue, and exercise intolerance could all be thought of as possible signs or symptoms of suboptimal riboflavin status. There's also some data suggesting that in pregnancy, preeclampsia, which is a condition of increased blood pressure, urinary protein, and edema during pregnancy, and which is quite serious because 5% of women who have preeclampsia progress to eclampsia, which involves seizures, hemorrhage, and potentially the mother and the baby can die. Women with preeclampsia, women who have biochemical evidence of poor riboflavin status have 4.7 times the risk of preeclampsia. So you could argue that preeclampsia is potentially a manifestation of suboptimal riboflavin status as well. So what do we know about riboflavin that might explain why all these things can go wrong? Let's start out talking about what riboflavin is and what it does in the body. So the word riboflavin comes from ribo, meaning that it contains the sugar ribose, which we also find in DNA, in RNA, and in all the major energy carriers that are involved in energy metabolism. And then it has the term flavin. And flavin comes from the Latin meaning yellow and refers to a group of closely structurally related chemicals called flavins. One of the key features of flavins is that they easily gain or lose electrons. To gain an electron is to become reduced. To lose an electron is to become oxidized. And these reactions where we transfer electrons are called redox reactions for reduction oxidation. And when we think of what we're trying to do with energy metabolism, we are basically breaking apart the molecules in our food and harnessing the energy contained in their chemical bonds. Those chemical bonds are in the form of shared electrons. So something that has the ability to gain or lose electrons is something that has the ability to rip apart food molecules and harness the energy contained in the chemical bonds. And that's why riboflavin is important to energy metabolism. Flavins are yellow when they're in the oxidized state and colorless in the reduced state. And that's why they derive their name from flavin, from the Latin meaning yellow. If you've ever made whey at home from milk by separating the curds in whey, you may have noticed the whey over time as it separates becoming bright yellow. That's the presence of riboflavin. If you've ever taken a riboflavin supplement or you've taken a B complex supplement and you looked at your pee afterwards and saw that it was neon yellow, that's the riboflavin. Flavins are also fluorescent. Riboflavin absorbs ultraviolet light, violet light, and blue light, and it emits green or yellow light in response. The fact that flavins are fluorescent is part of what allows us to quantify them inside the laboratory, because we can use detectors based on shooting light at the molecules and watching the light that bounce back. That's called a fluorescence detector, and that's what we use to detect riboflavin in chemical samples. Flavins are also photoreducible. That means that outside of our own energy metabolism, where we have enzymes saying exactly what they're going to, deciding exactly what to do with riboflavin to take which electrons from which molecules in food, if light strikes riboflavin, that can cause it to develop an excited state where it wants to grab an electron from something else in order to hold down the energy that came in from that light wave. Sometimes the flavins will reduce themselves, and that means that part of the molecule will bind to some other part of the molecule. Or sometimes they might oxidize something else nearby. This underlies quite a few implications. So first of all, this can lead to the destruction of riboflavin, and it can also lead to the destruction of whatever compounds they oxidize or grab the electrons from. So as an example of how this is leveraged in medicine, in newborns with jaundice with elevated bilirubin, they use phototherapy. And what happens in the phototherapy is that the light strikes riboflavin, causes it to get into an excited state and oxidize the bilirubin that's there in excess. This helps them clear the bilirubin, but it also destroys the riboflavin in the process. And so the infants that this phototherapy is used for develop riboflavin deficiency as a result and need to be properly supplemented in order to control that. Riboflavin is also used in eye surgery for keratoconus, which is the weakening of the cornea. What they do here is they peel off the cornea and part of the layer of cells underneath, and then they load it with riboflavin, and then they blast it with light. And what happens is that the riboflavin, when it's hit by the light, will then oxidize collagen and cause the collagen to cross-link more, which strengthens the collagen. And this has the result of strengthening the cornea and correcting the weakening that was otherwise taking place. Riboflavin also degrades into riboflavin analogs that are inert. They don't have any activity as a vitamin, but they can compete with riboflavin for absorption and utilization. That's not just true with humans, it's also true with fungi. And so riboflavin can be used in treatment of fungal keratitis. So this is a fungal infection in the skin that is inflaming the skin. And the antifungal drug amphotericin B makes the fungi have more porous membranes, and that allows things to get in that otherwise wouldn't into the fungal cell. And the phototherapy destroys riboflavin in the skin, creates these riboflavin analogs that act as riboflavin antagonists, and then get into the fungal cells and basically poison them by antagonizing riboflavin in their metabolism. The ability of light to interact with riboflavin also underlies why light can destroy the riboflavin in foods. The main reason why commercial milk went from glass bottles or clear plastic to cartons was because in a clear container, the blue light or ultraviolet light can destroy the riboflavin in the milk. Okay, so these basic characteristics of fluorescence, redox reactivity, and photoreducibility are general properties of flavins. When we take a modified flavin molecule and connect it to ribose, we get riboflavin. And riboflavin is very structurally similar to the chemicals that make up DNA. So if you've taken a biology class, you may remember it or you may have forgotten it, but you definitely learned about how DNA is a language made up of 4 letters. And each of these letters consists of a nitrogenous base that is connected to a ribose molecule and then connected to a phosphate molecule. So to take as an example, adenine is a nitrogenous base that has ribose added to it to make adenosine, and then it has phosphate added to it to make adenosine 5-phosphate or adenosine monophosphate, AMP. Although riboflavin is not a part of DNA, it's structurally similar enough to the components of DNA that it does qualify as a nucleoside and a nucleotide. So the flavin molecule is a nitrogenous base like adenine. The riboflavin molecule is a nucleoside like adenosine. And riboflavin 5-phosphate is a nucleotide like AMP. Riboflavin 5-phosphate is also called flavin mononucleotide or FMN for that reason. If you see supplements, you may see some supplements called riboflavin 5-phosphate. You may see other supplements called flavin mononucleotide or FMN. These are the exact same thing. Flavin mononucleotide can join together with AMP, the other nucleotide, and make FAD, flavin adenine dinucleotide. FMN and FAD are the 2 flavocoenzymes that we use in our body in all of our enzyme systems. When they bind to enzymes, together we call these flavoproteins. So that's the basic chemistry. Now let's take a look at the biochemistry to help us understand what are the roles that riboflavin is playing in the body. There was a 2013 paper that identified 77 known flavoproteins, although I would call those known or suspected. 23 of them were just based on inferring that they probably use riboflavin based on their structure. So the actual pool of known flavoproteins is smaller than that, but suffice it to say that riboflavin plays a lot of roles, and there are still we still have a lot more to learn about what riboflavin does in the body. But there are a handful of roles that are very, very well established, and those are its roles in energy metabolism, in the antioxidant system, in methylation, and in detoxification, and other nutrient interactions, such as interactions with vitamin B6, niacin, and iron, and a handful of others that I'll add into this. But let's really focus first on energy metabolism, because this is really the foundation of riboflavin's ability to support all these other systems. Really, everything in the body that we do requires energy, and everything that riboflavin does relies back in some way on what riboflavin does in energy metabolism. So we can think of riboflavin very much like the way that we think of niacin, which we'll cover in depth in the next podcast in this series. Riboflavin and niacin are both carriers of electrons, and this gives them their importance to energy metabolism because when we metabolize food for energy, we are ripping apart the food molecules and we're harvesting the energy contained in the chemical bonds made from shared electrons. In the case of riboflavin, we have FMN and FAD. Overwhelmingly, we're talking about FAD here. There are a couple of uses of FMN, but 84% of flavoenzymes use FAD instead of FMN. And in energy metabolism, we will look at one important use of FMN, but we're overwhelmingly looking at FAD. For niacin, we have a carrier very much like FAD, but it's called NAD. There are a couple of important differences between FAD and NAD. FAD is less stable in its redox state. The electrons that it holds onto, it's easier for FAD to give them up. FAD is more promiscuous than NAD. The range of reactions that it might get itself into is much larger than NAD. And so FAD is a liability if you're going to let it freely diffuse around. But it's an advantage if it's protein bound, because when a protein binds to FAD, it can stretch the FAD and it can twist the FAD and it can hold on to it more or less tightly to have a profound influence on exactly what that FAD is going to engage with for a specific chemical reaction, and also influence how tightly does it hold on to those electrons. So FAD has this huge range of things that it can do when it's bound to certain proteins, whereas NAD is a diffusible carrier. It's not protein bound. It's very well matched to be the thing that just runs around inside of the cell freely to bring electrons from one place to another. So what we find is that quite often FAD and NAD are both involved in the same reaction, but FAD is bound to the protein and is participating very intimately in transforming whatever that enzyme is doing. Like that enzyme is going to break apart glucose. FAD is very intimately involved in breaking apart the glucose, whereas the NAD is going to come up and is going to bring electrons with it from somewhere else, or is going to come up to that reaction and take the electrons out of it and go somewhere else with them. So FAD is transforming things, NAD is shuttling things around. Depending on the reaction, we will often see FAD passing electrons to NAD or the opposite of that. So let's take a bird's eye view of what's happening in energy metabolism in order to understand the role of riboflavin and in order to contextualize that role. And while we're going through this, let's think about why it would make a difference whether we're consuming carbs, protein, or fat, because these different macronutrients all affect the riboflavin requirement differently. So the bird's eye view is that all the food molecules we consume are contained of carbons. And the ultimate general fate that consuming protein or carbs or fat has when we're metabolizing them for energy is to be broken down into a 2-carbon unit called an acetyl group that will be carried using coenzyme A, which is made from vitamin B5, the subject of another podcast like this, to the citric acid cycle. And the purpose of the citric acid cycle is to rip apart that 2-carbon molecule, to release each carbon as carbon dioxide, and to harness the energy in the chemical bonds by bringing the electrons to the electron transport chain. The electron transport chain takes those electrons that had been in the food molecule and it passes them on through a series of dozens of different steps so that each step, a little bit of energy is released from the electron, and that energy can be used and harnessed to synthesize ATP. The carriers of electrons from the citric acid cycle to the electron transport chain are NAD and FAD. If we're burning carbohydrate for energy, we are going to rip apart a glucose molecule in glycolysis into 2 molecules of pyruvate. A glucose molecule has 6 carbons, Pyruvate has 3, so it's half of a glucose molecule. And then we're going to take one of those carbons off and release it as carbon dioxide to produce our acetyl group. If we're burning fat, we're going to use a process called beta oxidation. Different fats will be composed of different numbers of carbons, but the process of beta oxidation is going to rip apart those fatty acids 2 carbons at a time. Each time we'll make an acetyl group that can then go enter the citric acid cycle. Each time we engage in any of these things, splitting glucose into pyruvate, taking off the carbon for carbon dioxide to make the acetyl group, or splitting apart the fatty acid molecule 2 carbons at a time, all of those processes are releasing a little bit of energy. And that energy, just like the energy released in the citric acid cycle, has to get carried to the electron transport chain. And the carriers are going to be either NAD or FAD in some form. If we're burning protein for energy, things are a little bit more complicated. Different amino acids have different metabolic pathways, and all of the amino acids are at some point going to deliver something to the citric acid cycle. It might be acetyl-CoA, or it might be something else that goes into the citric acid cycle. It's so complicated that we don't need to get into the details here. But the citric acid cycle is the point at which everything shares in common. The citric acid cycle itself uses riboflavin twice for each turn of the cycle. For each acetyl group that we produce from glucose, after we break the glucose molecule in half to generate pyruvate, when we take off 1 carbon to release as carbon dioxide, we use riboflavin there a 3rd time. So for every turn of the citric acid cycle, when we burn carbohydrate, we use riboflavin 3 times. For fat, we're using riboflavin at a much higher rate. So we have the 2 riboflavin that we use for the citric acid cycle, but every time that we rip a 2-carbon unit off of a fatty acid to bring it to the citric acid cycle, we use riboflavin 3 times. So for every turn of the citric acid cycle, we're using fat. When we burn fat, we're using riboflavin 5 times. And when we use carbohydrate, when we burn carbohydrate, we're using riboflavin 3 times. On top of this, when we burn fat for energy, we are producing slightly less ATP per turn of the citric acid cycle than when we burn carbohydrate for energy. So if we adjust this for the amount of ATP generated, burning fat requires 89% more riboflavin than burning the same amount of energy from carbohydrate. That means that burning fat almost doubles the riboflavin requirement compared to burning carbohydrate. If we look at protein, it's more complicated. There are some amino acids that add to the riboflavin requirement, and that's mainly the branched chain amino acids, or BCAAs, and lysine, and to a much lesser extent, tryptophan. But when you look at the total number of amino acids, a lot of the amino acids in your dietary protein actually use less riboflavin if they're just going straight into the citric acid cycle. And so it averages out that protein uses even less riboflavin than carbohydrate does. But on the other hand, what you're primarily doing with protein is you're using it for structural purposes. And although you do use protein for energy, it's never going to be your main energy source. So what we would expect from the biochemistry is that the diet that maximally spares riboflavin would be a diet that has fairly high protein and fairly high carbohydrate, but is very low in fat. Now, that doesn't mean that you have to eat a low-fat, high-protein, high-carbohydrate diet. It just means that you would expect to need more riboflavin if you are eating a higher-fat diet, and you would expect to need less riboflavin if you are eating a diet that's based more on protein and carbohydrate. In addition, we would also expect that energy expenditure in general would raise the riboflavin requirement, and that riboflavin deficiency would hurt our ability to use energy. So we'd expect it to cause fatigue and exercise intolerance. And we would expect that if we are exercising a lot, or if we have a high metabolic rate, we're going to need more riboflavin. We would also expect that if you do poorly on a high-fat diet and you feel a lot better on a high-protein or high-carbohydrate diet, then suboptimal riboflavin status might be part of the reason. And then finally, if you look at how the different types of exercise affect how you burn energy, then we would expect that low and moderate intensity endurance exercise, which primarily burns fat, would raise the riboflavin requirement more than high intensity exercise, which burns less fat and relies more on creatine and carbohydrate, if you adjust for the total workload. Now, it's kind of hard to judge the total workload. So you could very easily wind up burning more total energy by doing high intensity work, and that might make you need more riboflavin because of the higher energy expenditure. But if you were to control for the energy expenditure, I would expect, based on the biochemistry, that it's the low and moderate intensity endurance exercise that would be raising the riboflavin requirement. Closely related to the system of energy metabolism is the antioxidant system. The antioxidant system is designed to protect our tissues from wear and tear and to protect our metabolism from becoming dysregulated. It's protecting against oxidants. Oxidants can go by various names like free radicals or reactive oxygen species or even reactive nitrogen species. What they share in common is that they are hungry for electrons and they can damage our tissues or damage our metabolism by going around and, in their hunger for electrons, taking them away from other molecules. Oxidants actually play valuable roles in regulating our metabolism. The antioxidant system is not designed to bring oxidation down to zero. It's just designed to protect our cells from damage and to make sure that oxidants are used for their good purposes, but don't cause harm. And to do that in a very controlled fashion, they need to neutralize those oxidants by giving them the electrons that they're hungry for. And in this system, we can see a few ways of it operating. For example, imagine that vitamin E is in our cell membrane, and it is trying to protect the fatty acids in the cell membrane from damage. What vitamin E has to do is deliver an electron to the oxidant and neutralize it. Where does vitamin E get that electron? It gets it from vitamin C. Where does vitamin C get that electron? It gets it from glutathione, the master antioxidant of the cell. But where does glutathione get that electron? Glutathione gets that electron from riboflavin in the enzyme glutathione reductase. Riboflavin got that electron from niacin. Niacin got it from glucose. So in this system, we are taking electrons from our food molecules, just like we're doing in the system of energy metabolism. But instead of harnessing the energy to make ATP, we're harnessing those electrons to neutralize oxidants and protect them from causing damage. As such, the Fa D that is part of the glutathione reductase molecule represents the interface between the system of energy metabolism and the antioxidant system. So what we see here is the system of energy metabolism is fundamentally the foundation of the antioxidant system. And one of the things that we'll see is that if you are deficient in riboflavin, it appears that your defense against oxidative stress in the antioxidant system is one of the things that suffers first. And we can imagine why that might be, which is that, first of all, you use the antioxidant system to invest in long-term protection of your tissues and long-term health goals. Whereas if you don't have energy, all of the processes you need for immediate survival in your body will fail right now. And so they're much more imminently important to your survival. But also, if riboflavin's participation in antioxidant defense is directly relying on its participation in energy metabolism, then riboflavin doesn't support energy metabolism first, its role in the antioxidant system becomes completely useless. So one of the things that we would expect to see from this biochemistry is that before you reach a point where your ability to extract energy from food is compromised, you'll see greater wear and tear on your tissues because of oxidative damage first. A third system that riboflavin supports is the methylation system. Methylation is very important to your mental and physical health. If you want to learn more about it, go to chrismasterjohnphd.com/methylation. The role of riboflavin in methylation is that Fa D is part of the MTHFR protein. MTHFR is the enzyme that produces methylfolate, and riboflavin is providing the electrons, again taken from niacin, taken from glucose, just as in the antioxidant system, and it's using them to construct the methyl group of methylfolate. Riboflavin is also involved in recycling glycine. So one of the things that happens in the methylation system is that if you have too many methyl groups, you use the amino acid glycine as a methyl buffer. Then you try to recycle that glycine. In order to recycle that glycine, you need to use riboflavin. Riboflavin also is involved in using trimethylglycine, or TMG, also known as betaine, which you can either get a supplement called TMG, or you can make this from choline inside your body. Riboflavin is involved in the second step of using TMG. So if you use TMG as a methyl donor, it loses a methyl group and becomes a dimethylglycine. In order to extract the next methyl group from that dimethylglycine, you need to use riboflavin. If riboflavin deficiency is hurting your ability to extract methyl groups or to synthesize methylfolate, we would expect to see elevated homocysteine as a possible result. And Alex will talk about some studies on homocysteine later. And we would expect to see many of the signs and symptoms that we would otherwise associate with deficiencies of folate, when in actuality, the problem might be that our folate intake is fine, but we're not utilizing the folate properly because we can't make the methyl group because of the vitamin B2 deficiency. Riboflavin also has some interactions with other nutrients. So riboflavin is needed to convert the form of vitamin B6 that is found in plant foods and in a lot of different supplements called pyridoxine into the biologically active form pyridoxal. There are some supplements that are pyridoxal 5-phosphate, or P5P. This is not relevant to those supplements. Additionally, when you consume vitamin B6 from animal foods, you consume pyridoxal and pyridoxamine. This is not relevant to consuming B6 from animal foods. But if you are relying on plant foods or on pyridoxine hydrochloride supplements for your vitamin B6, then you need to have sufficient riboflavin to convert that pyridoxine into the active form. Riboflavin is also involved in the synthesis of niacin from tryptophan. Tryptophan is an amino acid that we get from our protein. We convert tryptophan to niacin using riboflavin, also using iron, and also using vitamin B6. We also consume niacin in the diet. So in order to become deficient in niacin, you probably have to be deficient both in dietary niacin and also in any one of the following: protein, because it supplies the tryptophan; riboflavin, iron, or vitamin B6, because it helps convert the tryptophan to niacin. In other words, if you just don't have much dietary niacin, you probably won't become niacin deficient because you will be able to get the niacin from tryptophan. And if you're just riboflavin deficient, you might not become niacin deficient because you might have niacin in your diet. It's when you're deficient in both the dietary niacin and one of those other factors that are involved in synthesizing endogenously that you would wind up causing a niacin deficiency. So But if you do have evidence of niacin deficiency, then you definitely want to look at your riboflavin status as a possible contribution. The mechanisms by which riboflavin affects iron status are not clearly worked out. However, riboflavin deficient animals have impairments in both their ability to absorb iron and to utilize it for hemoglobin synthesis. And there are a subset of people with iron deficiency anemia who only respond to iron when you also correct the riboflavin deficiency. So there are a number of people out there who can't get their iron up, even with iron supplements. And this comes back to one of the stories that I fabricated in the beginning of Jacinta, who had iron deficiency anemia, and she improved her iron status only after eating liver. Why? Well, because as we'll see, liver is the best source of riboflavin. And in that case, in that story, the riboflavin was what was helping her fix her iron status. Riboflavin is also involved in detoxification. There are a lot of papers out there, reviews, even recent reviews saying that riboflavin is important for the cytochrome P450 system, which is a family of enzymes involved in detoxifying drugs and environmental toxins. As far as I can tell from reading the literature, this point of view became outdated decades ago. And although riboflavin is not involved in the cytochrome P450 system, it is involved in a closely related system called the flavin-containing monooxygenases. These are 5 enzymes in your liver that carry out the first in several steps of the detoxification of a number of different drugs and toxins. These include FMO3, which is the enzyme that metabolizes trimethylamine to trimethylamine oxide, TMAO. This is the molecule that is infamous for having for being argued to play a role in heart disease when derived from carnitine and choline in your diet by your gut bacteria. TMAO is also found very abundantly in fish, and if you've been following my work for a while, one of the things that you've seen me argue is that I will not be very convinced that TMAO is causing heart disease based on deriving it from red meat and eggs in the diet until someone explains why fish doesn't seem to be associated with heart disease when fish is leading to much more TMAO. But the production of TMAO is just one of the things that is done by that family of enzymes, and they play a role in detoxifying a number of other compounds. There are a number of other riboflavin-dependent enzymes that I won't go into, but I'll mention a handful here. Riboflavin is needed for NADPH oxidase, which is a key component of the inflammatory response. There are some harmful roles of NADPH oxidase in chronic inflammation, but NADPH oxidase is also absolutely critical to your immune defense because you use the production of oxidants by that enzyme to kill pathogens. NADPH oxidase is also critical to the production of thyroid hormone. In your thyroid gland, you use it to generate hydrogen peroxide that you need to make thyroid hormone. Riboflavin is also a cofactor for monoamine oxidase, which is abbreviated MAO and plays an important role in modulating your neurotransmitters. And then finally, riboflavin is used for the enzyme that causes your proteins to fold correctly. Specifically, riboflavin is used to make disulfide bonds in those proteins, which is basically, if you imagine a protein as a long string, you need to shape it into a 3-dimensional shape in order for it to carry out its effects. And you need to glue it into that shape. And it's sulfur-sulfur bonds inside the protein string that help glue it into that shape. And riboflavin is used to make those bonds. And so one of the things that we'll get to in this podcast is the use of high-dose riboflavin supplements in order to help correct misfolded proteins in a variety of diseases. So let's look at the biochemistry. Let's talk a little bit about the physiology. How does riboflavin get from the food into us and fulfill those functions? If we look at riboflavin in food, most riboflavin in food is Fa D and FMN. In milk, it's free riboflavin. In fortified foods, it's free riboflavin. And in many supplements, but not all, it's free riboflavin. You will also see supplements of riboflavin 5-phosphate or flavin mononucleotide, FMN. Those are the same thing. When we consume food, we first start breaking apart the FAD and FMN from the proteins that it's bound to with gastric acid, stomach acid, and protein digestion. That is required to free the FMN and FAD from those proteins in the food. And if anything is hurting our stomach acid or protein digestion, that's going to hurt our ability to extract riboflavin from those foods. FAD and FMN are then hydrolyzed to free riboflavin in the intestine, which is absorbed usually in the proximal small intestine, meaning up toward the very beginning of the small intestine. And the absorption appears to be completely in the form of free riboflavin. Now, one of the things that I went into this podcast believing that I've changed my mind about is that it could be beneficial to take supplements in the coenzyme form. So I don't know of any supplements of FAD, but there are supplements of flavin mononucleotide and others of the exact same chemical called riboflavin 5-phosphate. And I had previously thought that if you have any kind of impairment in making FAD and FMN from riboflavin, that it would be beneficial to take those supplements. But based on what I've seen, I do not believe that it is likely that we absorb any amount of FMN or FAD intact. If there is absorption, it's probably not very meaningful, and it's probably like slipping between the cells. And so although there's in a person with a healthy digestive system, the FAD and FMN are completely hydrolyzed down to free riboflavin and are absorbed very well. If anything, I think that people with small intestinal pathologies, where the intestinal cells that are producing the enzymes that break FAD and FMN down to free riboflavin are damaged, I think in those cases, the free riboflavin, which is the form you get from milk, fortified foods, and the cheaper supplements, is probably going to be better absorbed. After the free riboflavin is absorbed into the intestinal cell, a portion of it is converted to FMN and FAD. Doing that requires ATP and it requires magnesium. And it's regulated by thyroid hormone and adrenal hormones. So if your energy status is low, if you're magnesium deficient, or if you're hypothyroid or hypoadrenal, you can have low conversion of riboflavin to FAD and FMN. The conversion of riboflavin to FAD and FMN helps lock it into the cell because FAD and FMN don't cross the cell membrane easily. In the intestinal cell, some of that will get bound to flavoproteins for this intestinal cell's own purposes. The bulk of it will be hydrolyzed back to free riboflavin, transported through the portal vein into the liver as free riboflavin, and then the liver will take its share. The liver, in proportion to your energy status and your thyroid status, your adrenal status, and your magnesium status, will convert the free riboflavin to FMN and FAD and lock it into flavoproteins. A portion of that will then circulate in the blood. Most of what circulates in the blood is FMN and FAD bound to proteins in the blood, mainly to albumin, but also to immunoglobulins. It appears that the proteins binding the riboflavin in the blood are part of what prevents riboflavin from spilling out into the urine. So if you have a high-dose riboflavin supplement, probably what's happening is the riboflavin is being retained by the proteins in your blood, and when you saturate that capacity to retain it, the excess free riboflavin is going to spill into the urine. It would seem to me, and I can't find any studies on this, but it would seem to me that a high-protein diet or diet that, or really anything that's going to impact the level of proteins in your blood is not only going to allow you to retain more riboflavin and prevent urinary loss, but it's also going to be a factor in the distribution of riboflavin to tissues outside your liver. So I think when your protein levels are lower, that's probably going to bias riboflavin towards being concentrated in the liver. And when your protein levels are higher, I think that's going to bias riboflavin toward being distributed to other tissues. In pregnancy, there's an increased production of proteins that can bind riboflavin in the blood, and that's part of what helps mobilize riboflavin to be delivered across the placenta. This also persists during lactation, where you want increased riboflavin delivery to the milk. When women are lactating, they will nourish the infant at the expense of their own bodies, but they will also have less of an ability to do so. So in a very deficient East Indian population, it was measured that 9% of the mothers were riboflavin deficient, 9% of the mothers had adequate riboflavin status, and 36% of their infants had adequate riboflavin status. In other words, these deficient mothers were producing a greater number of sufficient infants, and that's because the mothers would nourish the infants at their own expense. However, the amount of riboflavin in their milk was 30% lower than what you would see in people on an adequate diet. So their ability to nourish their infants was low as a result of riboflavin deficiency, but they still extracted whatever they could from themselves to deliver it to the infants, to give their infants a better riboflavin status than even they had. Once we understand the biochemistry and physiology of riboflavin, it makes sense to talk about how do we look, what do we use for a marker of nutritional status? And the reason is that we want to use this both to understand how to know if we have enough, but we also want to understand how to look for a good marker of nutritional status in order to interpret the research that's out there. Because the research needs to define how much we need, and it has to use a marker of nutritional status to do that. So the gold standard marker of nutritional status is the erythrocyte glutathione reductase activity coefficient, or which I don't know, it's abbreviated EGRAC, so I'll call it EGRAC. And what this is it's taking your red blood cells and it's looking at the enzyme glutathione reductase. So this is the enzyme that supports the antioxidant system by recycling glutathione using riboflavin. I mentioned before that we would expect the antioxidant system to be sacrificed before the system of energy metabolism. And the reason I said was that we need the energy metabolism system to make the antioxidant system work. In other words, glutathione reductase means nothing if you can't harness energy from your food. And we also it's much more imminent to our immediate survival to be able to produce ATP. So what we see is that EGRAC, the erythrocyte glutathione reductase activity coefficient, is a very sensitive marker of glutathione status that is very well of riboflavin status is very well correlated with riboflavin intake and very well defined against where do you start seeing clinical deficiency symptoms arising. But it's isolated to research. So there are many studies using EGRAC, and there is no commercially available test for it. The EGRAC is different than just measuring erythrocyte glutathione reductase activity. So in the testing nutritional status, the ultimate cheat sheet, I had mentioned the company HDRI that offers erythrocyte glutathione reductase activity as a marker of riboflavin status. I am very likely to revise that recommendation and take it out in the next edition of the cheat sheet on the basis of the research that I've been doing for this podcast. So the EGRAC, the coefficient, what they do is they take your red blood cells and they measure glutathione reductase activity with and without adding riboflavin in the form of FAD. The logic is that if you are riboflavin deficient, you will be producing that enzyme in a defective form that doesn't work properly because you don't have enough Fa D to act as its cofactor. This is a much more specific test of riboflavin deficiency than just looking at erythrocyte glutathione reductase activity. The activity just says they just measure what's the activity when we take out your red blood cells. They don't add riboflavin and see how much does adding riboflavin stimulate the activity. And erythrocyte glutathione reductase activity can be regulated by many other things. So, for example, if you're exposed to a lot of oxidants, then the activity goes up. If you eat a lot of vegetables, the activity goes up. There are things that can alter that, and you're not going to see the uniformity of the response to riboflavin status in just looking at the activity as you would when looking at the EGRAC, the coefficient, the question of how much does adding riboflavin stimulate that enzyme. There are 14 other biomarkers of riboflavin status that have been identified, but most of them have only been looked at in 1 or 2 studies, whereas EGRAC has been looked at in over 15 studies and has been characterized very well. So we will see disagreements on exactly where the cutoff should be, but what we're doing here is we're saying let's divide the activity with riboflavin by the activity without riboflavin and use it as a ratio. If there's 20% stimulation of the enzyme with riboflavin, then the EGRAC would be 1.2. If there's 60% increase with adding riboflavin to the assay, then the EGRAC would be 1.6. And there's general agreement that under 1.2 is adequate, whereas 1.2 to 1.4 might be considered low, and then greater than 1.4 might be considered deficient. But exactly where the line of inadequacy and deficiency is varies depending on who you ask, which researchers you ask. And some people argue that you should be looking more at symptoms. Some people argue that you should just take the mean of a healthy population and go 2 standard deviations above and below and take a normal range, and so on. But one thing that I wanted to do was see where can I get the closest equivalent to the EGRAC in a commercially available test. And my preliminary judgment is as follows. If you are going to get a commercially available test in the United States for riboflavin status, I think LabCorp's whole blood riboflavin is the best test. Quest's test of plasma riboflavin is useless and should not be used because plasma riboflavin is going to be highly vulnerable to your recent intake and is not going to be a reliable marker of long-term status. The problem is, I don't know how to correlate whole blood riboflavin to all the studies on EGRAC. However, I do know that erythrocyte riboflavin normalized to the concentration of hemoglobin in your blood does correlate very well with the EGRAC. And so I have a formula that I'm going to recommend preliminarily, and I'll put the details into the show notes here. But the principle is that when you get the whole blood riboflavin, you should also get a CBC from the same blood draw so that you have a hemoglobin measurement. You can assume that about 85% or 80 to 85%, so let's say 82.5% is an average of the whole blood riboflavin is in your erythrocytes. And then you can do a calculation to see what is your estimated amount of riboflavin per gram of hemoglobin in your blood. So I'll put the way to calculate that in the show notes. So check out the show notes if you want those details. And I'll also be updating the Testing Nutritional Status Ultimate cheat sheet with this. In the Great Plains Organic Acids test, the OATS test, they have glutaric acid as a urinary marker of riboflavin deficiency. I do agree that glutaric acid elevated in the urine would often indicate riboflavin deficiency, but it just isn't specific to riboflavin because there are other nutrient deficiencies that can elevate it, like thiamine and niacin, and there are genetic factors that can elevate it, like a deficiency in the relevant enzymes. So I think if you are using the Great Plains Oats test as a nutritional screening, I do think you could use that to say if your glutaric acid is elevated, you should further investigate your riboflavin status, but I would not use it to confirm riboflavin deficiency. When you're measuring your riboflavin status, you should do it after an overnight fast. Any riboflavin that you had from the evening before should be mostly gone in 5 hours and should be completely gone by 10 hours. So an overnight fast, I do not think that your results are going to risk being affected by riboflavin from the day before. But they should be fasting, and you should not take any supplements that contain riboflavin the morning of. There's been a lot of concern about whether taking biotin interferes with certain tests. It does interfere with certain tests, but it would not interfere specifically with the whole blood riboflavin test. If you are getting other tests along with it, it would be wise in general to stop any biotin supplements for about 3 or 4 days before the testing, just in case one of those tests biotin would interfere with it. Okay, now let's take this concept of the marker of nutritional status and use it to say how much riboflavin do we need to A, prevent deficiency, and then B, optimize our health. So the EGRAC is the marker that's been most used. And we should keep in mind when we're looking at this that EGRAC in these studies is used as a marker of nutritional status, but it isn't just a marker of nutritional status. It also represents the degree to which your riboflavin intake is optimized to support your antioxidant defense, which is likely one of the most sensitive things to riboflavin intake and reflects your ability to long-term optimize your protection of your tissues from wear and tear and to protect your metabolism from becoming damaged and dysregulated. So optimizing the EGRAC is a goal in itself, not just a marker of to what degree are we protected from having the classical signs of riboflavin deficiency. The RDA for riboflavin was set in 1998, which was a long time ago. At this point, this is we have decades of newer research, but it still is a foundation of what we use to think about how much riboflavin that we need. So we should look at how they came up with it. The riboflavin requirement for infants is set based on the concentration of riboflavin in the milk of mothers who are not deficient, and then what is the volume of milk that those mothers then give to those infants that the infants consume. And the RDA winds up being set at 0.3 milligrams per day for the first 6 months of life, and then it goes up to 0.4 milligrams for the second 6 months of life. Then the RDA was set for adults, and for everyone ages 1 through 18, it was adjusted backwards on body weight. So for adults, what was observed was 0.5 to 0.6 milligrams per day is the cutoff point where below that you start seeing classical clinical deficiency signs and symptoms. Above that, you tend to not see those signs and symptoms. Then, on the other hand, optimizing the EGRAC, suppressing it down to the normal level that suggestive of adequacy, was seen with 1.3 milligrams per day. So what they did for the RDA was they said, all right, we're going to estimate that the average person needs 1.1 milligrams per day if they're a man and 0.9 milligrams per day if they're a woman. Then we will assume, since we don't know what the variation is, we'll assume that one standard deviation is 10% away from that mean. And we want to add 2 standard deviations to protect 97 to 98% of the people from Deficiency. And so to take an estimated average requirement of 1.1 milligrams per day for men and 0.9 for women and add 20% to that gives an RDA, a recommended dietary allowance, of 1.1 milligrams per day for women and 1.3 milligrams per day for men. For people of ages 1 through 18, this was adjusted down for body weight. So for both genders, the RDA increases from 0.4 milligrams for the second 6 months of life. For the next 3 years, it goes up to 0.5. For 4 to 8 year olds, it goes up to 0.6. And then it diverges for boys and girls. So for boys 9 to 13 years old, it goes up to 0.9. And then for 14 to 18 year olds, it goes up to 1.3, which is the same as adult men. And then for girls, ages 9 to 13, it goes up to 0.9. And then it goes up to 1.0 for ages 14 to 18, all adjusted on body weight. My suspicion is that the adjustment for body weight is completely dubious because riboflavin is needed for your energy metabolism. And children have a high rate, like many of these age groups have a very high rate of energy metabolism and growth. And so I think it's very possible that their riboflavin requirements are much greater than what you would expect from adjusting an adult dose backwards for body weight. And indeed, we'll look at some data later that suggests that actually the proportion of adolescents actually that have biochemical evidence of riboflavin deficiency is actually very high. In pregnancy, they assumed that what they looked at was pregnant women showed a higher need for riboflavin both to prevent clinical signs of deficiency and to optimize the egg rack. And so that wound up adding 0.3 milligrams per day to yield 1.4 milligrams per day for pregnancy. And then for lactation, the RDA basically said, well, we're going to say that you're adding 0.3 milligrams of riboflavin into your milk every day and that you're about 70% efficient. In other words, when you add 0.3 milligrams per day, you're actually losing another 0.1 milligrams. And so adding 0.4 milligrams per day brought the RDA up in lactation to 1.6 milligrams per day. Alex, I'd like to turn it over to you here. So we are looking at the principle of the RDA being we want to bring the egg rack down to normal. And, normal has been defined differently in different studies. But we could say that what we really want to do is maximally suppress the egg rack. In other words, we want to make sure that the amount of riboflavin that we have to optimize our glutathione recycling and antioxidant defense is going to be maximal. It's going to be whatever will maximize our ability to support that. And so I know that you've looked at some of the studies of supplementation. Do you have a sense of how much riboflavin do you need when you combine the background diet plus any supplements that were added in the studies that you looked at to maximally suppress the egg rack?
Yeah, so when you look at what few studies there are in adults and there's more in children, when they enter into these studies, most people have egg racks that we would consider to be deficient. So they're starting out with levels that are around 1.3 to 1.4, despite consuming riboflavin that would be in line with the RDA, if not greater. And most people in industrialized countries consume adequate riboflavin according to the RDA. It's not considered a nutrient of concern. Yet they have these elevated egg racks that respond to further supplementation, usually with doses between 1 and 2 milligrams per day. And we do see further suppression of the egg rack when they do that, which brings their total food plus supplement dose of riboflavin per day up to around 4 to 5 milligrams. Now, unfortunately, we don't have any studies that directly compare the suppressive effects of doses of supplemental riboflavin that are around 5 milligrams, 10 milligrams, and up higher. We do have one study in the elderly that suggests that taking a supplement of riboflavin providing 1.6 milligrams suppresses the egg rack down to 1.2 milligrams, suppresses the egg rack down to 1.2, and that supplemental dose is on top of an already adequate diet. But if you give them 25 milligrams of riboflavin, we see further suppression down to 1.07 to 1.08. I haven't come across anything that shows suppression all the way down to 1, but it does appear that going as high as 25 milligrams provides the most suppression that we've seen in the studies. Whether getting further suppression from 1.2 down to below 1.1 would offer a benefit remains speculation, since we're within that normal range, but it certainly seems to be the case that the more riboflavin you can get, the better off you would be in terms of suppressing the egg rack.
So, Alex, we're talking, one of the things that I find a little bit hard to wrap my mind around is the idea that we need 25 milligrams to optimize the egg rack. So like, you can't get 25 milligrams from food. I'll go into food sources soon. But two things come to mind for me here. One is, the first is with the studies, I think we don't have enough data points, right? So like, comparing 25 milligrams to what was the comparison? Was it to 1.6 milligrams? There's a huge gap there, right? And then did you get any sense that there was any other studies that like compared doses that were smaller intervals between them?
No, I hadn't come across any studies. The best that we can do is there was a meta-analysis that was conducted that aggregated data from adults, children, and pregnant women, and it found the greatest suppression of the egg rack with daily dosing of 2 to 5 milligrams, which even beat out daily high dosing of 25 milligrams or more, although that later value is just based on 2 very small studies. One of which involved anemic men that could have played a role. But so far, the data suggests that higher doses are going to have a more pronounced effect on suppressing egg rack in, at the very least, the 2 to 5 milligram range. And that is, again, on top of what you're already getting in your diet.
Right. Okay. So then let's... actually, the second thing that I was thinking of was if you look at the physiology, you are trapping riboflavin in your cells in proportion to your energy status. And so this is dependent on magnesium, it's dependent on ATP, it's dependent on thyroid hormone, it's dependent on adrenal hormone. I think it's quite possible that many people who have suboptimal status for any of those things might need like an extra push of what seems like an unreasonable dose of riboflavin that you wouldn't get from food because their efficiency of trapping it is small, right? So imagine that the free riboflavin is coming in, some of it is getting locked, and then whatever is not getting locked in the cell because of low energy status or low magnesium status is basically just spilling over into the urine. And why is it happening? Well, one reason it's happening is because it's free riboflavin. You don't use free riboflavin. What are you going to do with it? But the other reason it's happening is free riboflavin is an oxidative liability. It's free riboflavin that can get damaged by light. It's free riboflavin that can oxidize other things and create free radicals and create damage. So you really don't want free riboflavin around unless you've converted to FMN and FAD and you've converted into flavoproteins. So if your status of magnesium or thyroid or adrenal or whatever is a little bit low and your efficiency of trapping it is like really low after you get up to some basic level of adequacy, then you can't really think about it like all that yellow stuff going into my urine is wasting. It's sort of like you might have to waste a bunch in your urine in order to harness the next 0.5 milligram out of your diet and put it to use. Like, does that make any sense to you?
Yeah, it definitely does, especially when you do consider what nutrients of concern in industrialized countries are. Magnesium is a big one, where over half the population does not have a sufficient intake, at least in America. And so those things can definitely play a role. And that may very well also explain why we see people entering into these studies with egg racks that would be considered insufficient or deficient in riboflavin despite consuming the RDA. Because the RDAs for every nutrient are based on studies that have been conducted in healthy individuals. And so whenever you start to mess with their health status or a diet that is not replete in all of the nutrients they require, then you can start to move away from the RDA and it will not always be applicable. I think this is a great example of that.
So I think that on the one hand, that provides a basis for trying to understand why we would need, why something like 25 milligrams might be helpful to someone. At the same time, I think if we just think about where this is found in dietary sources, it does make sense that a high quality ancestral diet, especially one emphasizing animal foods, would provide you the 2, 3, 4, 5, 6 milligrams of riboflavin a day that most of us are not getting. So let's take a look at these dietary sources. The way that I would conceive of this, and to be honest here, like the data's kind of all over the map. You could definitely, I'm trying to simplify this, but if you go into a nutritional database, you're going to find a big mess, right? But my way of simplifying this is to say that we could basically make 5 tiers of foods in terms of how good they are for providing riboflavin. And the very top of this tier 1, is liver. It's any liver. It doesn't matter if it's from a pig, a chicken, a cow, a lamb, or whatever. Liver is the top tier. With that said, lamb liver appears to be the best, and that lamb liver is better when the liver is imported from New Zealand than when it is from the United States. I don't know why, but what I do know is that the diet of grass-fed animals is going to produce better riboflavin than the diet of grain-fed animals. But riboflavin is usually added to the diets of grain-fed animals, and so it's, that's going to even it out. On the other hand, for ruminants, and lamb is a ruminant, for all the red meat animals, riboflavin is largely produced by the rumen. And so ruminants tend to have good riboflavin status no matter what they were eating because of the rumen production. Nevertheless, the diet can alter the rumen. So it could be that grain-fed animals that are supplemented with riboflavin, still because of the effect of the grain on the rumen microorganisms, have poorer riboflavin status than animals feeding on pasture. There might be a certain extent, especially in a ruminant, where whatever vitamins are in the feed are way less important than what the fibers and other constituents in the food are doing to the rumen in that animal. So the individual livers, probably the highest quality liver from the grass-fed animal is the best. And probably that's what we're seeing in New Zealand imported liver of a lamb being the best product. But still, across the board, liver ranks 2 to 5 milligrams for every 100 grams of food, or 3.5 ounces. So here's your, you were talking about adding 5 milligrams to the diet, like one serving of liver adds your 5 milligrams to the diet. In the second tier, we have kidney and heart. I don't know about every organ meat, but like kidney and heart, probably representative of many organs. Kidney, heart, and almonds rank in at 1 to 2 milligrams of riboflavin. That's tier 2. Tier 3 has red meat. It has cheese, but not milk. It has eggs. It has salmon, but not most other fish. It has mushrooms. Maybe not all the mushrooms, but many of the mushrooms are in this tier. Much of the seaweed is in this tier. Sesame, whether it's tahini or sesame seeds. Wheat germ and wheat bran are in tier 3. So basically, 1 or 2 servings of tier 1 per week can do a lot for your riboflavin status. 1 or 2 servings of tier 2, kidney, heart, and almonds, can do quite a bit for your riboflavin status. But if you had a serving every day, it would do, a lot for your riboflavin status. Tier 3, the red meat, cheese, eggs, salmon, mushrooms, seaweed, sesame, wheat germ, and wheat bran, you really have to work those foods into your diet on a daily basis to really drive up your riboflavin. You need multiple servings a day from this tier to drive up your riboflavin. In tier 4, we would put all the other meats. So if you're eating a lot of chicken, it contributes a lot to your riboflavin, just not red meat. You have to eat these are high volume to get a lot of riboflavin. So the other meats and milk I would put in here, they rank in at 0.2 to 0.4 milligrams of riboflavin. Tier 5 is most of the other foods that aren't junk. So whole grains, nuts, seeds, vegetables, beans, starches, most other foods that weren't mentioned here, if they are not unenriched white flour or sugar or fat, then they are in tier 5. They're doing something for your riboflavin status, but they don't really matter unless you consume a lot of them. Then besides these, we have flour products that are enriched. Flour products have been enriched since the 1940s, basically compensating for a diet that was poor in animal products. Like the riboflavin, it's not, in a sense, like we developed nutritional deficiencies when everyone started eating white flour and white sugar, but the riboflavin in the flour products is less compensating from the riboflavin that's gone from the whole wheat and more compensating for just not having liver, not having kidney, not having heart, not having red meat, not having cheese, not having egg, not having salmon. Now, in most of these foods, you are getting Fa D and FMN, but in the milk, you have free riboflavin. In the fortified flours, you have free riboflavin. And in many supplements, you have free riboflavin. My suspicion is that and I mentioned this before, that people with small intestinal pathologies that really hurt digestion are probably going to do best with milk, fortified flour. I'm not advocate for fortified flours, but I think they'll do best with milk, fortified flours, and free riboflavin supplements than they will from the good foods, simply because even though the good foods are better, they require more to digest and absorb that riboflavin. Many people think that nutritional yeast is a rich source of riboflavin. Well, if you look up Bob's nutritional yeast, you'll see it has a lot of riboflavin. Why? You look in the ingredients, because they added riboflavin. If you look at an unenriched nutritional yeast product, you're looking at getting 2% of the RDA of riboflavin for every couple tablespoons. So nutritional yeast is not naturally a good source of riboflavin. For most food processing, there's little to no effect. Even for soaking beans, there's very little effect. Cooking, very little effect. When you cook and you leach the cooking water out is when you see loss of riboflavin. So, for example, if you're cooking beans and throwing the water away, you're going to lose riboflavin. What I would say is that probably in the context of beans, it's kind of like, who cares? If you're relying on beans for your riboflavin, then there's other parts of your diet that are really missing. But if you are relying on beans for your riboflavin, probably what you want to do is any of the processing that makes them more digestible, you want to do prior to cooking. And then when you do finally cook them, so throw away the soaking water, but then when you do finally cook them, don't throw away the cooking water because if that means doing more pre-soaking and more throwing away the soaking water to make them digestible, then so be it. But you don't want to cook them in and throw away the cooking water, because that's when you start to lose riboflavin. But the overwhelming big way you lose riboflavin is from light. And unfortunately, almost all the studies I could find are on, in terms of how much light does it take to destroy how much riboflavin, are on milk and pasta. And my suspicion is that milk and pasta are much more vulnerable because they contain free riboflavin. And in meat, I think meat probably has much less loss because the proteins will protect the riboflavin. Nevertheless, what do we know from milk and pasta? Well, you're talking about, in milk, 2 hours of sunlight destroys half of the riboflavin. If you agitate that food less, you will get less destruction because the destruction mostly occurs at the surface. So if you really wanted to destroy the riboflavin, then leave the milk in a glass jar in the sunlight and then every once in a while shake it. That's going to move all the riboflavin that you haven't destroyed from the middle or the bottom into the surfaces and make it more exposed to light. But nevertheless, like just leaving the milk in a glass bottle in the sun for 2 hours will destroy half the riboflavin. Some other foods, some of the pasta foods might take a couple days being exposed to light to destroy the riboflavin, but still. So without having data on meat where I suspect the riboflavin is more stable, I would still choose to avoid exposing my food to light. And this ties back to the story of Jane, the story I fabricated at the beginning about Jane who changed her name to Clover, who was out exposed to light, tanning, and only drinking kefir that she was fermenting out in the light to absorb the light energy. If you're going to ferment kefir, you should do it in a bottle that either you cover it with an opaque cloth or you do it in an amber bottle or something to protect it from light. You definitely don't want it sitting out in the sunlight. Anything that's sitting out in the sunlight is going to destroy the riboflavin. And tanning was losing riboflavin. So I'll make this point in a few minutes again, but riboflavin is also destroyed when you are exposed to sunlight. In addition to considering the dietary sources, there also is riboflavin produced in the large intestine, in the colon. We know that we can absorb it. We know that the amount we can absorb is fairly large based on studies where they just directly infused riboflavin into the colon. That riboflavin doesn't get absorbed. No one has ever done the studies to say, to what degree does this contribute to systemic status? So it probably at least nourishes the colon cells, and it might nourish you. If it does, then I think that raises a giant question of all the studies on the riboflavin requirement, how much was that affected by their microbiome? Like, Alex, was there any studies that you saw that looked at the microbiome contribution?
Nope. The microbiome is already a very underinvestigated thing when it comes to, well, most everything. So, no, there's no studies looking at the microbiome riboflavin interaction that I'm aware of.
So one thing that we do know is that a microbiome that's more Bacteroides dominant has the bacteria that make up that microbiome. This was called enterotype 1 in one of the papers that came out. The bacteria in that microbiome have the genes for riboflavin synthesis more than bacteria in other microbiomes. But we don't actually know that they contribute more to our riboflavin status. We know that a vegetable diet produces more riboflavin in the gut than a meat diet does, but we don't know whether you actually wind up with better riboflavin status. Probably you don't, because the actual riboflavin in your food is mostly contained in the meat versus the vegetables. But it would be reasonable to say that you would get better riboflavin status if you eat a lot of riboflavin in your foods and you also eat the fermentable fibers and vegetables that nourish your gut bacteria for them to produce riboflavin. It's just a gray area that we don't know too much about. So from this perspective, like, how do we get good riboflavin status? Well, we eat from the top. We eat liver once or twice a week. We try to work in organ meats as much as we can. Maybe that means once or twice a week we eat kidneys. Once or twice a week we eat heart. And then we eat, more from the top tier, more from like the third tier foods than from the fourth and fifth tier foods. We also protect our foods from light. What are other things that we could think about as causes of suboptimal riboflavin status or deficiency? Well, this is in addition to just not eating the foods that are rich in riboflavin, what could we say? Well, to start with, not eating many of the high tier foods is fairly common, right? There's no organ meats in most people's diet. There's very little consumption. People will avoid red meat for health reasons. Maybe most of the people who do eat red meat are eating high sugar, which has no riboflavin, or eating high fat, or eating French fries, where there's like a huge amount of calories that provide no riboflavin. In the developing world, the deficiency is thought to be mostly associated with low animal product consumption. But you can imagine people who are health conscious who move away from refined, fortified grains out of health concerns. Refined grains, I'm not a fan, but they're a major source of riboflavin in the standard diet. If you move away from refined grains and you move toward foods that you consider healthy, like coconut oil, that have no riboflavin, you could experience a decline in riboflavin status. So if you're, I can imagine health conscious people moving away from refined grains and toward a higher fat diet. I would expect their riboflavin status to decline unless they're making up for it by adding more of the top tier foods like organ meats. Data indicate that exercise compared to being sedentary raises the riboflavin requirement by about 20%. Exercise is healthy, but it means you need more riboflavin. Light therapy to treat jaundice in infants works by the light hitting the riboflavin to generate oxidative stress that oxidizes the bilirubin that's present in excess and help get rid of it. As I mentioned in the beginning, those infants wind up with riboflavin deficiency as a result of this process. I haven't found studies on it, but I suspect that regular use of tanning beds, phototherapy for psoriasis, and deliberately seeking out sunlight for health-conscious efforts like circadian rhythm and vitamin D is all going to decrease riboflavin status. I'm not saying don't do those things. I'm saying that when you do those things, you might now need more dietary riboflavin than you needed before. As I mentioned previously, thyroid and adrenal hormones are involved in converting the riboflavin to FNN and FAD. So if you have hypothyroidism, where you have low adrenal production, that's going to impair, from a functional perspective, it's going to impair your riboflavin utilization. Alcohol impairs the absorption and utilization of riboflavin. I mentioned in the fabricated story of Jack, I mentioned that he was out drinking for 3 weeks and didn't eat any food. I actually pulled that concept right out of an old case report of riboflavin deficiency. In fact, let me quote it. This is a quote from one of the early papers on riboflavin deficiency from the Journal of the American Medical Association, 1939. Case 1, JAM are his initials. A white man, aged 34, was admitted December 30th, 1938, in severe alcoholic excitement. He was known to have been on a prolonged debauch and to have eaten little for about 3 weeks. There was a fine scaly dermatitis of the nasomolar grooves, an ally of the nose as well as of the chin, etc., etc. So as a result of not eating as much riboflavin and then impairing the absorption and impairing the utilization, alcoholism is a cause of riboflavin deficiency. Anorexics rarely eat enough riboflavin, as with most of the other nutrients. Diabetes, stress, trauma, and dialysis treatment are all thought to increase the excretion of riboflavin. Diabetes, cancer, and heart disease can precipitate or exacerbate a deficiency. So if you just connect these two sentences, right? Diabetes, stress, and trauma. Well, the stress and trauma of disease states can increase your riboflavin turnover, and you go from marginal to deficient if you don't have adequate riboflavin status when those diseases really hit you. In the RDA report, they speculated that if we know that pregnancy and lactation increase riboflavin demands, then it's safe to assume that someone who's pregnant with multiple fetuses or someone who's nursing multiple infants is going to have even further increased riboflavin needs. It was once thought that oral contraceptives were a cause of riboflavin deficiency, but studies that looked at people on oral contraceptives where their riboflavin intake was controlled for didn't find any difference. So that's probably not a factor. Anything that hurts your stomach acid or your protein digestion is going to hurt your absorption of riboflavin from food. And so these digestive problems are actually digestive problems all the way down, all the way from the stomach to at least the proximal small intestine, maybe even into the colon, are going to hurt your riboflavin status. So in the stomach, it's hypochlorhydria, poor protein digestion that hurt your ability to extract riboflavin from the proteins in food. Then on top of that, we know that inflammation in the small intestine decreases the riboflavin transporters. So in inflammatory bowel disease, you have poor riboflavin status due to poor absorption due to inflammation. You could surmise that SIBO or anything going on in the small intestine that causes inflammation is going to hurt riboflavin status. And then in the large intestine, or maybe even the small intestine, if you have SIBO, you can have microbial. You can first have loss of the bacteria that make riboflavin. Maybe that is a factor. But also, bacteria in the gut can make the same degradation products of riboflavin as light does. So light converts riboflavin to lumiflavin and lumichrome. Bacteria degrade riboflavin, especially to lumichrome. And this has been shown in rats. It hasn't been shown in people, but probably it is a factor. And what that means is that you can have the wrong bacteria in your gut that are not only going to degrade the riboflavin, but they are going to produce riboflavin antagonists that hurt your riboflavin absorption. And then they do get absorbed. They hurt the riboflavin transport into cells. And then they do get into the cells and they hurt the riboflavin utilization inside the cell. So fixing the gut is going to be a major factor in fixing riboflavin status for a lot of people. And unfortunately, there's just very little known about how to optimize the gut for riboflavin status compared to what we know about you should stop drinking alcohol, you should eat riboflavin-rich foods. There are also a number of genetic defects in riboflavin metabolism. And these include a number of different mechanisms by which riboflavin transports its electrons into the electron transport chain. It includes the transporters that get riboflavin into the mitochondria. It includes even elements of the electron transport chain. So for example, there are genetic disorders in complex 1 of the electron transport chain that are riboflavin responsive. And that's probably because riboflavin as FMN, riboflavin doesn't just carry electrons to the electron transport chain. Riboflavin as FMN is part of complex 1. And that might be why some complex 1 disorders respond to riboflavin. There are also defects in the transporters that get riboflavin into the intestines for absorption and defects in the transporters that get riboflavin into the brain. And they all have some unique spin on how that manifests depending on exactly what's happening to the riboflavin. But many of these disorders, many of the times, most of these disorders, most of the time, can be treated with riboflavin at very high doses. So we're talking about doses of 10 milligrams per kilogram body weight. If you are an 150-pound person, you're about 70 kilograms. So we're talking 700 milligrams of riboflavin. The maximal dose used is 1.5 kilograms. Excuse me, not 1.5 kilograms. 1.5 grams total of riboflavin is the maximal dose used in any of these studies. And probably to some degree, you're overcoming a defective riboflavin transport. But studies are showing that the high-dose riboflavin helps take proteins that have been misfolded, that have been incorrectly put together, and helps correct how they are folded into their three-dimensional shape. And what's fascinating is, although we've mostly studied this as riboflavin correcting the folding of the riboflavin-dependent proteins, we also now know that riboflavin is part of the mechanism of how we fold all these proteins that need to be folded correctly. All the proteins that have disulfide bonds in them utilize riboflavin to be folded correctly. So there may be many other disorders involving dysfunctional proteins that aren't flavoproteins that do get benefit from high-dose riboflavin on the order of 1, 2, 3, 400, 500 milligrams. So looking at all these potential causes of poor riboflavin status, like how big of a problem is it? So Alex had mentioned earlier that there's quite widespread cases of people whose AGRAC scores get better when they are supplemented with riboflavin, even if they seem to be consuming enough. Well, let's look at some of this data here. So the genetic defects, obviously, those are very, very rare. But just looking at suboptimal status, well, usually when people look at America or Canada or the United Kingdom or any of the other developed countries, it looks on the surface like no one's really riboflavin deficient. No one is reporting many cases of clinical symptoms of the severe deficiency, although no one's really looking for it either. But just looking at intakes, everything looks fine. So the average intake in the United States is 2.1 milligrams per day. That's higher than the RDA. If you look at how that is spread out, 10% consume less than 1.3 milligrams per day. That is pretty good, right? Because 90% of people are consuming more than the RDA for an adult man. And the RDA is supposed to support the needs of 97 to 98% of the people. And so, how many people who are consuming less than that are, actually need 1.3 milligrams? Like maybe the rate of suboptimal status when you look at that data is only 5% of people. And yet so the assumption here is that in the developed world, only pockets of people with low socioeconomic status who are eating very poor diets have riboflavin deficiency at a population level, unless we have very specific individuals who, for example, maybe if they were an alcoholic and they weren't eating for 3 weeks, maybe that person's going to develop a riboflavin deficiency. But on a population level, you're just not going to see it. And yet, there was a study in the United Kingdom that looked at the prevalence of an AGRAC score over 1.3, which is thought to indicate low suboptimal status of riboflavin. And they found that 66% of adults between the ages of 19 and 64 had AGRAC scores indicating poor riboflavin status. And when they looked at that separated by age, it was 41% of adults aged over 65, and it was 75% among boys aged 4 to 18 and 87% among girls aged 4 to 18. When I look at this, I see a couple things. First of all, if we take this data at face value, and why not, suboptimal riboflavin status is very common. Second of all, what I see is a major flaw in the logic of adjusting the RDA downward for body weight. And granted, the people who made the RDA did not have access to this data in 1998 when they made it. But the principle was that if an adult needs 1.3 milligrams, then a child weighing half as much as that adult weighs should need half that amount. When you think about what riboflavin is doing, though, you might say, geez, these people who are growing a lot, they need a lot of riboflavin because they're dumping energy into that growing, right? Like the teenage boy or the teenage girl, when they're in their growth spurt, how much are they eating? Right? They're not eating less. They're not eating half the amount of food as an adult who's twice their body weight is eating. They're eating more because it's so energy intensive. So I think that probably the RDA underestimates what we need to optimize AGRAC scores. And I think like Alex was saying before, it's probably something more like 2, 4, 5, 6 milligrams. But also, I think teenagers and growing children, probably they need, even more riboflavin than the adults do, is my suspicion. Alex, why don't you take over from here and tell us a little bit about what you found from supplements? So, like, who should be supplementing? How much should they be supplementing? What has it been shown to be good for? Does the form matter? What can you tell us about supplementing with riboflavin?
Okay, So unfortunately, like Chris mentioned earlier, none of you are going to have access to an egg rack test, which would be very easy to just tell you whether or not you should consider supplementation, right? But there are certain conditions where if you have them, it would be worth it to consider riboflavin supplementation. And one of these would be iron deficiency anemia. Now, there isn't much research in this area, but what is available suggests that taking, in particular, in one study, 10 milligrams per day in men who had iron deficiency anemia was able to increase hemoglobin from 106 grams per liter up to 125 grams per liter, keeping in mind that the lower limit of normal is 130. So these iron anemic individuals were able to almost get above the lower limit for anemia simply by supplementing 10 milligrams of riboflavin per day for 2 weeks. These benefits were seen in 14 days and they were maintained for another 2 weeks after that. So if you have iron deficiency anemia, then, depending on what your diet looks like and working with your physician and all that, it would be worthwhile to consider adding riboflavin into any other supplements like iron that you may be taking. Another condition where you're going to want to consider supplementation is going to be based on your particular polymorphism for the MTHFR mutation. And this relates back to cardiovascular health through its impact on homocysteine levels, which is an independent risk factor for heart disease, right? So we have quite a bit of data suggesting that the relationship between riboflavin and homocysteine is mediated by the MTHFR phenotype. So if you're one of the people with the normal CC allele or the CT allele, then it's unlikely that riboflavin is going to play a role. However, if you are homozygous mutated for this enzyme, then we see that riboflavin supplementation with doses again around the RDA are able to reduce homocysteine quite significantly, as much as 20% and upwards of 40% if you also happen to have an egg rack above 1.3. So this has to do with the way that the MTHFR gene or enzyme binds to FAD. When it's acting normally with the wild type CC genotype, then it's going to bind to the FAD perfectly fine. However, when you have the mutation towards the TT homozygous genotype, then you're going to have a reduced affinity of this enzyme to FAD. And that's going to cause reduced enzyme activity because MTHFR needs that riboflavin in order to function properly. So this would explain why supplementing with riboflavin would provide a benefit in these circumstances, because you're going to simply overcome the reduced affinity by saturating it with more riboflavin.
So I find this particularly fascinating for a couple of reasons. One is that I'm even in my mind right now, I'm revising my own protocol for methylation, which, I've always mentioned that these other B vitamins are important for MTHFR, but I've always kind of put it in the background. Whereas if what this paper is arguing about the FAD binding is true, then we might, we shouldn't take it for granted that your MTHFR is just lower because of these polymorphisms. We might actually be able to make that MTHFR either normal or a lot closer to normal, and maybe riboflavin should be the first line of defense. So I've been poking holes in other people's methylation protocols by saying, so the worst methylation protocol in the world, or the worst MTHFR protocol, in my mind, is to blast your cells with more methylfolate. Because yes, methylfolate's important, And yes, there's a place in my protocol for it. But methylfolate actually shuts down the MTHFR enzyme, like from, because you don't need it anymore if you're putting the methylfolate in. And so it's just much more to the root of the issue to use the riboflavin.
Yeah, and what you're saying right there is actually supported by one study in particular. And this was a recent one just done in the last couple years that took a bunch of people and compared their homocysteine values based on both their riboflavin status determined by the egg rack as well as their genotype for the MTHFR enzyme. And what they found is that people who were heterozygous or homozygous for the T mutation in the MTHFR enzyme had significantly higher homocysteine levels than people with the regular CC genotype only when the egg rack was above 1.2. There was no difference between genotypes when the egg rack was considered normal or optimal, below 1.2.
That's amazing. Also because, it's amazing just like not only to support the idea that this is only a problem when your riboflavin status is suboptimal, but it's also amazing to think that when we don't take riboflavin into account, we see this as a general effect of MTHFR, and that seems to support what we were saying before, that there's just widespread suboptimal riboflavin deficiency. In other words, all this body of research that shows this general effect of MTHFR, all of it is shown because it's all done in the background of a population that has widespread marginal riboflavin status.
Yeah, it's definitely a confounding variable that, at least to my knowledge, has never been taken into account. It's certainly something to consider. It would be really interesting to... what hasn't happened is, getting a really, really nice large-scale study using supplementation that is stratified by both the MTHFR genotype and riboflavin status. And it would be really cool to see how all of those interact in an actual controlled trial to affect a marker like homocysteine. Another area that can be affected by the same mutation is going to be blood pressure. And we have several studies suggesting that riboflavin may be effective at reducing it, at least among, again, people with certain mutations in the MTHFR enzyme. So this has been less well investigated than looking at homocysteine. But what data is available suggests that small doses of riboflavin supplementation at the close to the RDA of 1.6 milligrams are able to further reduce blood pressure more than medications would. Or I should say, if you add riboflavin in addition to blood pressure lowering medications, you get a greater effect at lowering blood pressure, but only in people who are homozygous for the T genotype. And I don't have an explanation for this, but it is possible it relates to homocysteine and the way that homocysteine interacts with endothelial cells within the artery walls. It is possible that these studies, the participants had lower homocysteine, like we've already seen, even though it wasn't measured in these studies. And then that lower homocysteine had a benefit for the functionality of the blood vessel walls that could have reduced their constriction. It could have made them more fluid and helped lower blood pressure additionally.
So I'll just jump in and say that, with the mechanism in MTHFR, there's homocysteine, but then there's also methylation itself. Like there could be dozens of things that are being methylated that maybe some of those tie into blood pressure. And then there's also glycine too, because actually glycine and choline, right? So when your MTHFR activity is lower, you are less effective at using using folate to methylate. So you rely more on choline and you start using up your choline more. And then also because methylfolate is the off switch for the glycine buffering system. And if you don't know what this means, go to chrismasterjohnphd.com/methylation, where I explain all that in detail. But when you have low methylfolate levels, you methylate glycine and you lose it in your urine. So you lose glycine that way too. So there's kind of a whole Pandora's box of different mechanisms that could wind up opening.
Yeah. So we have really two areas of research left. And the first one I want to touch on is exercise, because as Chris mentioned, exercise increases riboflavin requirements. And this makes sense because of its role in energy metabolism. So if you're going to go out running a lot, particularly if you're an endurance athlete, then you're going to require more riboflavin simply because you're going to be burning through more glucose and fatty acids. And so that is the entire background that first started the investigation into the effects of riboflavin supplementation on exercise performance and recovery. So when it comes to actual exercise performance, we have a bunch of studies in women, children, elite swimmers, distance runners that show no benefit on performance, not even with up to 60 milligrams of riboflavin per day. And this could be simply because energy metabolism is prioritized. So if these people were not getting adequate riboflavin, what they were getting would have been shuttled to energy metabolism and sustaining their athletic performance, rather than other important things like antioxidant function. And this gets into the one area of exercise research where riboflavin might have a benefit. It's far less investigated than it is for performance, but it's looking at the effects of high doses of riboflavin on muscle damage and soreness, which comes back to the antioxidant effects, since that muscle soreness you experience is the result of the inflammatory cascade from muscle damage and having to repair it. And we have at least one well-controlled study in really long-distance endurance runners. And I'm talking about like a 161-kilometer trek through the Sierra Nevada mountains, right? So what they did is they divided up all of the participants of this race randomly into one of two groups. And one of the groups consumed a placebo, and the other group consumed riboflavin, 100 milligrams, 30 to 60 minutes before they started the race, and then another 100 milligrams about halfway through the race around the 90-kilometer aid station. And what they found is that although riboflavin supplementation didn't reduce ratings of muscle pain or soreness or improve performance, like when considered on the race day as well as the several days following it, when they broke it up into the various sections in an exploratory analysis, they found that riboflavin did improve subjective feelings of recovery and reduce subjective feelings of muscle soreness from the athletes during the actual race. And that would make sense given these mechanisms. You're going to blast them with a high dose of riboflavin. You're already prioritizing energy metabolism, so performance isn't going to be affected. But now you're also giving riboflavin to support the antioxidant functions, which can help reduce muscle damage during this strenuous event and throughout it. Now, the question remains whether or not this effect is significant from a practical standpoint, because the difference between the groups was about 1 point on a 10-point scale, and the researchers determined ahead of time that they needed at least a 1.5 point reduction to be clinically significant. And so certainly we need more research on this, and there's a lot of speculation we can make about like potential long-term benefits of supporting the antioxidant system throughout consistent long-distance endurance training. It could result in, for example, less illness, which is something known to affect endurance runners quite significantly. But it is suggestive, and there is biological plausibility to speculate that it would have a benefit.
So I have a question that I know I'm inevitably going to be asked about. And I've given a little bit of thought to this, but I haven't read all the research, and I'm not sure what you might or might not have researched on this. But there's bound to be questions on, is that going to hurt your adaptive response to the exercise? In other words, do you require the oxidative stress and damage of the exercise in order to get the fitness benefit? And is feeling less discomfort during the exercise just mean that, less pain, less gain, right? What's your perspective on that?
Yeah, so we do have a bunch of studies that rather consistently suggest that taking high doses of vitamin C and vitamin E as the source of antioxidants do impair muscle growth, gains in strength, and even mitochondrial biogenesis from people performing resistance and endurance training exercise. But I don't think that we can extrapolate these to all antioxidants, because we have to consider how they're working in the body. When you blast yourself with high dose vitamin C and E, you are basically just flooding yourself with systemic antioxidants. But with riboflavin, it's only having that antioxidant effect when it gets transformed into its Fa D and FMN parts in the body, right? Free riboflavin by itself is actually an oxidant, and your body's going to flush it out with some lovely bright yellow pee as much as it can. So it's going to have antioxidant effects if it's able to get inside the cell and do its job. And that is probably going to be limited by what the body needs to deal with. So if you increase the amount of oxidative stress on your body, then your cells might be more responsive to taking up more riboflavin to help deal with that. But the body has those natural inborn feedback mechanisms to where it's not going to overwhelm itself with the antioxidant riboflavin, unlike the high dose vitamin C and E, which kind of overrides your body's natural feedback loops.
Yeah, I think that's a great way to look at it, and I would add a point to that, kind of riffing off what you're saying, that riboflavin, its antioxidant role is it doesn't just get converted to FAD, but that FAD has to become a cofactor of the glutathione reductase enzyme, the enzyme that's recycling glutathione. And what I think is interesting specifically in this respect is the oxidative stress of exercise or some other insult that is going to provoke a fitness response. Part of that, basically what you're trying to do is you're trying to burn more energy and you're trying to burn it cleanly. And so you make more mitochondria, and you also make more of all of the antioxidant defense system components that will allow those mitochondria to be protected during high rates of energy metabolism. And so part of the fitness response to the oxidative stress is to make more glutathione reductase. And that glutathione reductase can only do anything if it has the FAD necessary to be a cofactor. So you actually, I think here, you wouldn't be blunting the stimulus. You would actually be enabling the fitness response with the riboflavin.
Yeah, definitely. That's great insight. And the point you raised about the mitochondria, that's actually a great transition into the last area of research where riboflavin has been somewhat heavily investigated, and that has to do with migraines. So migraines can essentially be thought of as like the worst headache of your life ever, and you want to go die. And we don't know like what causes them specifically. There's a lot of hypotheses out there, and one of them has to do with impaired mitochondrial function in people who get migraines frequently, specifically defects in both the brain and mitochondrial energy metabolism. And so it's hypothesized with riboflavin as a precursor to FMN and FAD that riboflavin can help facilitate mitochondrial ATP production. And in this regard, a few studies have found that riboflavin is effective for reducing migraine frequency and duration when taken in ridiculously high doses of around 400 milligrams per day. There's one study that uses a lower dose of 25, a study that uses 50 milligrams, and a study that uses 200 milligrams, and none of those have shown a benefit. The only studies that have shown a benefit have used 400 milligrams per day, typically as one dose, although another study divided it up into 2 doses and taken with food. It would make sense to take these high doses with food, as that can increase the amount that you absorb, simply because riboflavin isn't completely soluble in water. And so when you form micelles in the small intestine by taking it with food, that will help get more of it into the body. And these reductions in headache frequency are seen within about 3 months. And the data for making their intensity less isn't as convincing. The studies either show no benefit or a small reduction of about 20%, although depending on your starting point, that could be a lot. But certainly the high doses reduce the frequency and duration with less certainty that they reduce the intensity.
So have you, in these papers that you've been looking at, what do they think is going on there? Why would the high doses be necessary? Do they speculate about the mechanism involved?
Yeah, so the speculation with the mechanism is simply that the defect in the mitochondria that is present in people who suffer from frequent migraines has to relate to the FAD and FMN ability, or I should say the components of the mitochondria, whereby taking super doses is going to flood the mitochondria and somewhat override this deficit that's naturally present. Similar to the MTHFR mutation, how taking more riboflavin can overcome the reduced affinity by saturating it with more of the substrate. So this would be a similar mechanism, but they haven't pinpointed exactly what it is.
Yeah, I think I think we will eventually see the expansion of problems that can be at least partly fixed with these high-dose riboflavin supplements. And I'm just guessing this based on 2 pieces of data. One is that in the riboflavin transport defects or defects in other riboflavin-dependent enzymes that produce severe symptoms that were in the past treated with these 100, 500 milligram doses of riboflavin, we know from those that the riboflavin is helping the protein fold correctly. And yet we also, in this same course of, that sort of opened up the expansion of the knowledge of that all protein folding in the endoplasmic reticulum, basically not all protein folding, but all disulfide formation that solidifies the 3-dimensional shape of probably the vast majority of proteins in some way or some respect, is all riboflavin dependent, right? So you can imagine scenarios where maybe the enzyme involved in doing the folding of everything has a higher affinity for FAD. And then you have like hundreds of of things that are fixed when you have the high dose riboflavin in there. Or maybe a defect in a protein is not an intrinsic defect, but it's a reduced, it's somehow, I should clarify, I don't know everything that's known about how this works. And I suspect that we only know a tiny bit collectively of how it works. But just, using my imagination, I can imagine that maybe a defect in a protein that causes it to be misfolded is that it has a lower affinity for the FAD that is stuck to the enzyme doing the folding of everything. But even though the FAD is part of that enzyme, it also has to interact with that protein while it's being folded. And so that protein's defect is that it has a reduced affinity for the FAD in the folding enzyme. And so somehow that plays into there. Like there's just so many different possibilities of what what potentially could go wrong and what could be fixed. But yeah, so there we have it. We have deficiency through suboptimal status through rationales for potential high dose supplementation. Let's go through a few short answer rapid fire questions where we can each weigh in on what I think inevitably are going to be practical questions that people will ask. And maybe we've covered them, but here we can kind of bullet point them out. So first of all, riboflavin, does it matter whether we get free riboflavin or riboflavin 5-phosphate or some other form that's out there?
No, the form shouldn't matter. So what research is available does suggest that most forms of riboflavin are going to be broken down in the intestinal barrier before it enters into your body. So whether you get it as free riboflavin or FMN or FAD, it's all going to be turned into free riboflavin when you absorb it. And then it will be retransformed after it's broken down back into the FMN and FAD as needed, particularly within the liver during that first pass by the liver.
Okay, so my speculation is that that's how it usually works, and that if anything, some people with small intestinal pathologies maybe don't break down the FAD and FMN from food, and so they probably might do better with free riboflavin than any of the other forms. Next question would be, actually, I don't know if you came across anything on this. Did you come across anything on sublingual riboflavin? No? Okay, neither have I. So that's out of there. Now, next question would be, we're taking it with food, right? You had just said that. So do we take it with food? Yes. Not an empty stomach. Then
it's necessary.
Okay, clarify.
Yeah, so I wouldn't say taking it with food is necessary. But taking it with food will help absorption, especially if you're going to be taking higher doses, like upwards of greater than 10 milligrams. So when you take it without food, the pharmacokinetic studies suggest that we can maximally absorb at any one time upwards of 25 milligrams. And I'm a little skeptical of this simply because these studies never tested super high doses like the 400 milligrams used for migraines, and there is some evidence that once the transport carriers or the transporters that work on riboflavin, once they get saturated, there is potential for paracellular absorption where it moves between the cells of the intestinal barrier to get absorbed into circulation. So that's a possibility with super high doses. But as far as doses upwards of 60 milligrams, maximal absorption occurs around 25, and the rest just you poop out. And when you get to doses above this, though, above the 25, then we start to see that food helps increase absorption and the retainment of riboflavin. So I would say that taking it with food certainly doesn't impair absorption, but the benefit would probably only be there if you're going to start taking really high doses.
So, this is based on small doses being absorbed really well in an empty stomach, or?
Yeah. So it's not just small doses. I mean, they do small doses, but it's graded pharmacokinetic studies that will use any like 1 milligram, 2 milligram, 5, and then 10, 25. And they work up to see how they all compare in terms of the serum response. What is important and needs to be mentioned here is that when they're looking at this, they aren't looking at the egg rack, how it's affected. They're just looking at how serum levels of riboflavin and all of that stuff are modified, since that's going to be the best marker of an acute riboflavin intake from a single meal.
Right. And so, presumably whatever helps you absorb the most in the long term helps you optimize the egg rack the best. So yeah, I mean, my guess here is that when you have it with food, your cells are in a higher energy state. ATP levels are higher because you have the influx of food, and ATP is what is retaining the riboflavin in the cell. And then probably your protein levels are higher, and protein synthesis levels are higher, so you're synthesizing the enzymes that bind the riboflavin. The protein levels in your blood that bind the riboflavin are probably higher and prevent it from going into the urine and so on. I think the next question we would get would be, does it matter if you, like, how often do you have to take riboflavin? And so I know that, I've been asked a question before, what do I think of Perfect Health diet recommendations to do, I forget exactly what the recommendations are, but non-daily dosing of the B vitamins. In this case, it seems a little bit of a gray area because you can kind of stock up on riboflavin if you can absorb 27 milligrams in a meal. But then again, how much do you retain of that? Because a lot of that gets left off in the urine. So have you seen anything indicating whether when you equalize for the total dose over a week, does it matter if you take it every day or not?
I have not come across a single study doing that, or a study that compares daily to bolus dosing. In fact, I'm only aware of one study that uses non-daily dosing, and it shows that there is still a benefit, but it's not nearly as strong as a benefit as you see with daily dosing. And the overwhelming majority of research uses daily dosing. So even though we could sit here and speculate about reasons why bolus dosing might be superior to daily, I see absolutely no reason why we should rely on speculation when we have a bunch of hard data that's actually done supplementation and shown effects and benefits with daily dosing of various amounts. I would just do daily.
Right. And then for the high dosing, like if someone's going to take 400 milligrams a day, have you seen anything on whether it matters if you take it once or separate it across multiple meals?
Yeah, most of those studies just use one big bolus where they have the participants consume 400 milligrams at once, although there is one that divided it into 200 milligrams twice per day. And I mean, presumably you could take also, I guess, like 50 milligrams 4 times per day. In theory, you would retain more of the riboflavin if you took more frequent lower doses because you wouldn't saturate your transport carriers in the intestine. But that hasn't been investigated.
Yeah, and I guess the last question around this out is: have you come across any adverse effects of riboflavin supplementation? I mean, we're talking here. Like I mentioned a case of 1.5 grams a day. You're talking about studies of 400 milligrams or 200 milligrams a day. This is massively over the RDA of 1.3 milligrams for the adult male. So, what, if anything, is the negative consequence of such high doses?
So, no upper limit for toxicity has been established for riboflavin. And so that's very telling, because that means from a toxicological standpoint, you don't need to worry about like getting cancer or any of that stuff. There is no upper limit that you need to be concerned with. In the literature, the most common side effect in the migraine literature from taking like 400 milligrams per day was some abdominal discomfort, and in one person, diarrhea. Logically, if you're going to mass dose something like that, then if you have a sensitive stomach, you could probably anticipate having some GI distress from it. But for the vast majority of people, it's, the only real side effect you'll notice is some very fluorescent urine.
Yeah. In everything that I've looked at, the only thing that I can find is speculation, reasonable speculation, but speculation that the ability of free riboflavin to generate free radicals, especially when exposed to light, is a potential risk. And my judgment on that is yes, but you generally don't retain much free riboflavin. Like, basically the system is designed so that you retain it as Fa D and FMN bound to proteins, or you pee it out. And probably there is some kind of negative effect if you do high dose riboflavin and then you go in a tanning bed when you have peak levels circulating like every day for years or something like that. You're probably putting more wear and stress on your body. But in terms of like, where does that fit in the context of every exposure to something stressful that your body faces? Like, I have no idea if that is actually meaningful or not. So I think that riboflavin should be considered non-toxic for all practical purposes. And yet at the same time, these 400, 500 milligram doses, it's like, if you have a good reason to do that and you get a benefit from it, then the speculation that there might be some harmful effect long term is just so speculative that it shouldn't outweigh whatever tangible benefit you get. But if you have no reason to do that, then it probably doesn't make sense to prophylactically take 500 milligrams of riboflavin a day, because now you have speculation versus speculation and you have nothing good to go on.
Yeah, I'll add to that too. If you are someone who's going to take a high dose, light exposure, if that's really the only concern, then it makes sense to take that high dose before you go to bed, because then you're not going to be exposed to light. And so you could, in fact, circumvent that speculative issue altogether, because you'll be in dark for the next, 8 hours, keeping in mind that riboflavin will be out of your system in like 5 to 10 hours.
Yeah, interesting thought. Okay, well, with that, let's wrap this up. Thank you, everyone, for listening this long, and we'll see you in the next episode. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes: 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com. Ancestral Supplements, putting back in what the modern world has left out. All right, everyone, I hope you enjoyed that. If you would like a transcript of this episode, you can get transcripts by becoming a member of the CMJ Masterpass. You can use the code Mastering Nutrition to get 10% off lifetime discount of this program, and you can do so by going to chrismasterjohnphd.com/masterpass/mastering nutrition. Using that link or the link by clicking on the link in the description of this episode, that will automatically apply the discount code so that you don't need to do any work to apply it. The CMJ Masterpass doesn't just give you transcripts, it also gives you ad-free content that comes out when it's produced instead of when it's released to the public. So you get pre-release content that is often weeks or months early, and you don't just get this for Mastering Nutrition, you also get it for my short practical tips in Chris Masterjohn Light. And although I'm not quite up to speed on Masterclass with Masterjohn yet, I will be at some point in the next couple months, and those will start coming out, and you will start getting them early inside the Masterpass. If you'd like to find more of Alex, you can find his work at alexleaf.com. And if you like more of me, you can find me at chrismasterjohnphd.com or at chrismasterjohn on any of the social media channels. Until next time,